about the TA schedule during exam week. So I sent that out in my announcement, like all the changes that we're going to make. We do it so that it makes like the most sense for you guys taking your exams to be like doing the TA session that corresponds with the exam that you're about to take. The only confusion I have is about genetics. Okay, well, we'll solve that. Okay. So I'm going to share my screen now. Give me one sec. Okay. So we're going to cover, you were right about Genome Browser. It's great. Yeah, it's like literally the bane of my existence, but it'll be okay. We'll get to it. What we're going to do is get through this PowerPoint today, and when he starts talking about Genome Browser, we're going to stop and we're going to cover that next time so that we can just do Genome Browser in its entirety and not break it up, kind of like what he did. Sometimes he just plans to go over a certain amount of material and then he talks so fast that he gets through that material and there's like leftover time and he's like, oh, well, why don't we just like get ahead? But we're not going to do that. We'll just do Genome Browser in its entirety next session. So today we're going to cover just this PowerPoint, the Human Genome 1. So like September 9th lecture and the 14th. Is there a lot of Genome Browser questions on the exam? Yes, there will be Genome Browser questions on the exam. And I did get some of those wrong on the first exam and they come back on the third exam. So I had to like, you know, get my shit together. Are they pictures? Yes, they are pictures. And they're going to be kind of blurry and you're just going to have to deal with it because he like screenshots them. So I guess like the quality kind of just goes down a little bit. I don't know, but they were not the best pictures. We just have to get through it. We just have to deal with it. I do want to say though, if you try to zoom in, sometimes Proctorio will kick you out. And I know if you're at home and it's your first exam and you zoom in and you get kicked out, you're going to probably panic. So I would just honestly try not to zoom in. Because both times I ever tried to zoom in on an exam, I got kicked out and I panicked. Okay. So with that being said, we're going to cover today September 9th lecture, September 14th lecture. So it's going to be this Human Genome 1 PowerPoint. We're going to stop when he gets into the Genome Browser. And we're going to do Genome Browser and then the Human Genome 2 next review session. And then the last review session, we're just going to have one PowerPoint, which is lecture 9. It's the instability of the human genome. He hasn't uploaded it yet, I don't think. And then there's one more article with highlights. So we'll cover that on the last TA session before your exam. And then I will have office hours before your exam so that you guys could just come ask me any questions. I can go over like some high yield stuff and yeah. So how should we study / prepare for Genome Browser? So we'll go over it. I'll go through it with you. I kind of just, the first time, the first exam, I kind of just didn't take it as seriously as I should have. Like I thought I understood it. My TA kind of like went over it a little bit, but I thought I got it. And I guess I just didn't get it. So I wouldn't just listen to me do it or listen to him do it. I would go into Genome Browser and see if you can like pick it out yourself, because that's what I did wrong on the first exam. But I fixed that for the last exam. So I didn't really miss those questions on the last exam. So listen, well, he already lectured, but if you want to go back and rewatch his lecture and then next TA session, listen to me go over it. And then after doing that, go over it like on your own. Okay, what happens if you get kicked out? Well, it prompts you to like click or something. I don't remember off the top of my head. You just like click to enter back and it lets you enter back. Will I be posting a practice exam? Yes. Like we said during the last Q&A session, all the TAs are going to give you a practice exam of 50 questions. Usually it's like the week before or something. I'm gonna work on yours this weekend. I know sometimes, like when we were in the program, our TAs would post the practice exam like 2 or 3 days before the exam, but I'll try to have it to you the week before your exam. Will it be online? Will what be online? Elizabeth, I'm not sure. Oh, my office hours. Oh, yes, it's going to be online because I can't, there's no room that's available to accommodate all of you guys if you all decide to show up. So just to keep it fair, I'm just going to log into Teams and I might make a separate link just for like a one-time office hours thing. And if I do that, I'll send it out in an announcement. And you guys could just click that link. It should go also to your calendars. And I'll just hang out there. When are my office hours? I guess I have them kind of set for September 29th, 6 to 8 p.m. and then September 30th, the morning of your exam from 9 to 11 for any last minute questions. And I'm always available through email for any questions. Will we have class after each exam? Yes, you will. So let's get started then. If there's no more questions, like housekeeping questions. Okay, here's our objectives. What time is your exam? It's at 2 p.m. If you log in not at 2 p.m., you're effed. You have to take it at 2 p.m. Okay, and that's 2 p.m. like Eastern time. So if you're in another state, make sure you know what the time change is and log in 2 p.m. Eastern time. Okay. So we have our dogma of molecular biology. We've talked about this before, from DNA to RNA to protein, and now we know that we can go from RNA back to DNA with reverse transcriptase, and he said that's one of his favorite enzymes. And then he says we have reverse transcriptase left over from just like random viruses that have inserted their genes into our genome, and it's just been left there, but it has no physiological function for us. It's just like leftover. Reverse transcriptase equals retrosynthesis. Retroviruses do use reverse transcriptase. I don't know if you could say that they're exactly equal. Okay, so we get into this a little bit more, this pre-initiation complex, like we discussed in a previous lecture. So TF2D, we know, is what kind of initiates everything for this pre-initiation complex. And it has like 12 subunits, but we don't need to know all 12 of them. The two important ones for us to go over right now are going to be the TBP, which is the TATA binding protein. And then the second one we already talked about, recognizes methylation of lysine 4 on histone 3. So those are the two important subunits that he wants you to know. So this first one is the new one. So the TATA binding protein, the TATA box is like where the primary transcript is going to begin. So it's 25 to 35 nucleotides downstream from the TATA box. That's when we start our primary transcript. So the TATA box is just like, okay, this is where we're going to start. It's going to recruit our RNA polymerase 2 and our TF2D, which is, initiating this whole pre-initiation complex, can recognize that it brings in our RNA polymerase 2 and, we get a transcription. So our RNA polymerase 2 is going to be used for protein coding genes. Okay, So here, our Tata box is an example of a regulatory sequence. He doesn't really like the word promoter. He likes regulatory sequence better. And then we already know he doesn't like antisense. He likes template. And coding is better. I put a line here because he likes coding strand better than sense strand. Okay, so here's our Tata box. We have our RNA polymerase moving in an uninterrupted fashion. So it's basically like the leading strand. It's just moving in an uninterrupted fashion. And then, so this is our DNA. So then now we're generating our primary RNA transcript. We have a 5' untranslated region and we have a 3' untranslated region. So now we have our introns and our exons in our primary RNA transcript, and then we get cleaving of those introns, and now we just have our mature mRNA, and then it gets translated into a polypeptide. So that's the sequence that's going on here. So downstream, like we talked about, is the direction of RNA polymerase. So if RNA polymerase is going to the right in this image, then that's going to be downstream. So like this last little box here would be downstream. And if we talked about like this promoter region, that's going to be upstream. And then primary transcripts are what we use when we're defining a gene space. So we can use that in Genome Browser and other bioinformatics. So it's mostly gets processed into mRNA, and we say mostly because introns get removed and exons are fused together. And then the exons are what's encoding the amino acids, which are making the proteins. And that's important because in cancer, some of our proteins are changed, so that's why we care about exons in medicine. Okay, so then here we see like a primary transcript, and he has primary transcripts in quotes because we see T's instead of U's. And we know that if we're working with RNA, we're going to have a U instead of a T. Oh, and then exons are in yellow here. So all of this is introns in between. Okay, so now that we have generated our mature mRNA, we're going to look at translating. So here's our RNA. Our 5' end corresponds with the N terminal of a peptide, and the 3' corresponds with the C terminal. So we have our start codon, AUG. Then we get our tRNAs loaded with our amino acids and their anticodons binding, and then we're growing a polypeptide. So mRNA goes out into the cytoplasm, gets translated into a protein. tRNA is what's bringing in the amino acids. Okay, so here, I'm pretty sure, if I'm remembering correctly, there was a question on our exam regarding something with this V600E mutation. I don't know if that's going to necessarily be the case for your exam, but this is an important topic to understand. So we know we have all of these different codes for our proteins. So we look at valine. At the position 600, if we're going from a glutamate in the BRAF protein, we know that glutamate is negatively charged. And when we want to turn on that BRAF protein and we want to proliferate, it gets phosphorylated. Phosphate has a negative charge, glutamate has a negative charge. So if we're going from valine to glutamate, then our protein is going to think it's being turned on. We're going to be proliferating, and that's like how you can get cancer. So if we look here, we have GUA versus GAA. So it's more likely if you have this sequence to get a mutation in one of the three And that's how you can change. So if you get a U to an A, then you're going to get this valine to glutamine mutation, versus if you have GUU that codes for your valine, then it's less likely because you're not going to get two mutations right next to each other. So to go from GUU to GAA is not going to be very likely. But if you go from this GUA to this GAA, that could be more likely. So you get that mutation. Your protein thinks the negative charge is similar to the negative charge on phosphate when it gets phosphorylated to turn on. So then we're just going to be proliferating, and we can get uncontrolled growth and cancer. So stop me if you have any questions on this. What is the BRAF protein? You're going to learn more about that later, but it's also found in cancer. What is BRAF protein? Yeah, so you're going to learn more about that later, but when it turns on, it tells us to proliferate. So if we have this mutation, or if we have another mutation in our BRAF protein and it just turned on, then we're just going to keep proliferating. That's all that's important for you to know at this time.
I have a question.
Okay.
So essentially you're saying that if we have the mutation that takes the GUA to GAA, then it normally would think that like with the negative charge would get phosphorylated, that would turn it on and then cause just excessive proliferation?
So normally without the mutation, the BRAF protein gets turned on by getting phosphorylated. So when it gets phosphorylated, those phosphates have a negative charge. So that's how it gets turned on. But with phosphorylation, you can phosphorylate and dephosphorylate to turn it on and off. So if we want to proliferate, then we can phosphorylate it. It proliferates. Okay, we're done now. We can take those phosphate groups off, and now we remove that negative charge. So, okay, it turns off. But if we have a mutation to a glutamate, then there's always going to be that negative charge. So the protein's kind of confused, like, oh, I'm phosphorylated, I have this negative charge, I need to just keep proliferating, and it's never going to be taken away, so it's not going to stop proliferating. Does that make sense?
Yes, thank you.
So I wrote it here.
And Phoebe, sorry, quick question. Valine is the one that normally gets proliferated to turn on proliferation. I mean, to turn on proliferation. Valine is the one that normally gets phosphorylated.
No, I'm not sure where the protein gets phosphorylated.
Okay.
But valine, it's just easy for this GUA to get mutated to this GAA. Yes.
Okay.
And I wrote it here for you guys that BRAF protein is just a pro-proliferative kinase. You're going to learn more about it later. All you need to know is that it's pro-proliferative.
I think he said in the chat or in class that there was like a serine 3 amino acids down that usually gets phosphorylated, to answer that question.
Okay, gotcha.
Thank you.
Good. Okay. You mentioned that because we know this mutation stimulates phosphorylation, we can assume it's phosphorylated somewhere nearby. Okay. But what's important to understand is that this glutamate has a negative charge, and it's a V600E mutation in the BRAF protein, which is a pro-proliferative kinase. Okay. So then we go into this definition of a gene, which we say we most often reference genes by their primary transcript. Sometimes it can be considered to begin at or close to the point where the DNA sequence, at or close to the point in the DNA sequence where transcription is initiated, and close to the end of where transcription terminates due to the consideration of regulatory elements. So it can be a little bit before and a little bit after this primary transcript, but we most often refer to genes by their primary transcript. So this slide is kind of pointless. Every nucleotide has a number in our genome. That's how we sequenced it with the Human Genome Project. So here's our DNA. We can have a gene A on this portion, a gene B on this portion, and a gene C here. Then if we have gene expression, we get proteins based on those genes. Does the primary transcript include the 5' and 3' UTRs? That's where we can kind of get into this or close to the point in the DNA sequence where transcription is initiated, because they're not translated. So we can say that they are. We see them here. That's our primary transcript. That's a 5' UTR, and that's a 3' UTR. It just depends on what you're referencing and who you're talking to, but yes, they can be included in the primary transcript. Okay.
Hey, Ivy, what does
Yes.
the UTR stand for?
Untranslated region.
Thank you.
Uh-huh. So here we have our chromosome in its mitotic conformation, composed of 2 double-stranded DNA molecules. So we have one sister chromatid here and another sister chromatid here. So if we're looking at only this portion, then that's 10% of the chromosome arm. We have like roughly 40 genes there. Then we look at this portion of it, 1% of the chromosome arm, and it only has 4 genes there. And then if we want to look at this specific gene, okay, here we have it, 1 gene, and we see like all of our exons, our introns, and then with gene expression, we'll get our RNA and then translated into a protein, and then the protein is folded. And that's how we express those genes. So that's just like the bigger picture of like what we're talking about. Okay, so this slide. Sorry, my words are a little small, but if you download it, you can zoom in and see them. But he doesn't really give us much space on these slides to write. So if we look at this top strand, the 5' to 3' strand, we see all these little boxes here. We see all these arrows. So if we look within this box he has for us boxed off, the bottom strand doesn't have any of those blue boxes. So those blue boxes are representing our exons, and then the little gray line in between is representing our introns. And these arrows are representing variable start sites. So if we want to express this gene, we can start transcription from this second arrow if we don't want these two. So you can tell which one is the coding strand and which one is the template strand by seeing which one has the boxes for exons. So in this case, the top is the coding strand, the bottom is the template strand, but either strand can be used as coding or template, depending on where the gene is located and what is being transcribed and translated. So if we look like here, for instance, right off to the right of this box, we can see that the bottom strand now has those blue boxes for exons, whereas the top strand doesn't. So then now the top strand would be the template and the bottom strand would be the coding. mRNA is the transcription product, which includes a 5' UTR and the 3' UTR in the mRNA transcript. These regions are not translated, though, to make the final product. That is how I understand it. You are correct. Is the top strand the minus strand? That's going to be more with Genome Browser, so we'll go into that next time. And I'll show you like specifically what to look for on Genome Browser. He's just trying to introduce the idea of coding versus template and how to determine that right now. So I'll go over that more with you guys when we talk about the Genome Browser. You're saying it depends on what region of the strand we're looking at to know which is the coding and which is the template. Yeah, so it depends on what gene that you want to transcribe and translate and express. So if I'm talking about this gene right here, this third little block right here, then if that's the gene I want, then my template strand is going to be the other strand. Are the arrows pointing in the direction of the transcription? Yes. So if I want to transcribe this gene, my arrow is going to be before it and then pointing like towards it. Okay. So then in this blue box, he just wants to highlight that there's a mistake because it's showing 2 exons overlapping, which we don't really ever have this, because the possibility that each of these exons has the same code for the same amino acid that can be used in the same protein is just very slim. The only example he has of this is the 3' UTR, and they don't even code for amino acids. So you're never going to really see that. He mentioned something about exons not overlapping. Yeah, that's what I'm talking about right now. So they're not going to overlap because we don't have that possibility of having the same code for the same amino acid in the same protein being functionally the same within these 2 overlapping amino acids, because we know that we're complementary. So that's just not going to work to have the same code for the same amino acid one way, the same code for the same amino acid the other way, and then have 2 different proteins that have the same amino acid at that position and have different functions. That's just not going to happen. And his take-home message on this slide for this bottom portion for figure B. So we see that we have like all these different RNAs, short RNAs, SNO-RNAs, ncRNAs, miRNAs, and then these antisense ncRNAs. You guys don't have to know all of these right now. He's going to go into them. I think it might be the last exam when we go into what all of these things are. But right now, what he wants you to know is that there can be other genes than just protein coding genes. Like these RNAs that don't code for proteins, but they have some other function. For example, miRNA regulates translation. SNO-RNAs regulate post-transcriptional modifications. And then this antisense ncRNA plays a role in shutting genes down and regulating the chromatin. So they don't specifically encode for proteins, but they have some other function. And we talk about that more later. Okay, so gene expression refers to the entire process whereby the information encoded in a particular gene is decoded into a particular functional protein. But non-coding RNAs have already been mentioned, and the development and specialization of tissues reflects the differential expression of genes. So these RNAs have some effect on expressing those genes. Okay, so now we're going to get into the different types of DNA sequences that we see. So we have single copy, moderately repetitive, and highly repetitive. So he's going to go in detail on these, and then he'll give you examples, so you should know the examples for each. So characteristics of single copy DNA sequences, we have 1 copy or a few copies per haploid genome. So it doesn't always have to be exactly 1 copy, but it's a few, a small number. There's no upper or lower size limit in length of like base pairs. And almost all of the human genes are in this category, which kind of makes sense because we just need 1 copy of most genes. Not all single copy sequences are coding regions or parts of genes. So a lot is just in between the genes connecting everything together. Okay, so here's the size of some of the human genes. This column to the left is what he wants you to memorize. So one of you asked me on Friday what the units are. So they're kilobases. You should just know that this number is kilobases. He's not going to try to trick you with that. He's pretty transparent. He just wants you to know these numbers and know what relationship the size has with like function and stuff. This may be a question for Dr. Blank about the overlapping exons and palindromic sequences. The palindromic sequences can explain why the exons. Yeah, I don't know the answer to that question, but what he was saying in class was that it's very unlikely to have two sequences that encode for the same protein and having that same protein at that position, or that encode for the same amino acid and having that same amino acid at the same position in a protein. It's just very unlikely. Okay. So memorize these numbers. He starts off by saying this globin gene, the HBB gene, is very small. It's 1.6 kilobases. You're welcome. It's 1.6 kilobases. It's on the small side. Then he says SRY is even smaller, 0.9. And then he says this dystrophin gene is really big. It's like the largest gene. And then we can see this cystic fibrosis is also pretty big. And we care about that because mutagenesis is random. So that means that globin is pretty well protected because it has a short number of nucleotides that can be altered. Whereas muscular dystrophy, which is a mutation in that dystrophin gene, is seen fairly common because the dystrophin gene is so large, it's like giving us so much space to acquire mutation. So it's a bigger target for mutation. So gene size dictates the target size for mutation. So the larger my gene is, the more likely I can have a random mutation in that gene. So more examples are RB, that's a very large gene, and we know RB is mutated in certain cancers. And then we have BRCA1, which a lot of people know about. So that's a huge gene. And if you already inherit one defective copy and then the other one gets mutated because it's such a large gene, it's a greater target for mutation, then throughout life, if you build up mutations in that one normal copy, then you're going to get breast cancer because you already inherited a defective copy and now you've already mutated your one good copy, so then that can lead to breast cancer. Do we need to know the human genes associated with their sizes? Yes. So that's why I have memorized and have them highlighted. Know those. He just wants you to know like the smaller ones and the larger ones. Like these middle ones, I don't remember being tested on those middle ones. Okay, now we get into this topic of gene families. So a gene family is just a set of genes that descended by duplication, and they're slight variations from that ancestral gene, but they're homologous throughout their entire sequence. And they may be clustered together or dispersed on different chromosomes. So an example of this would be not just knowing SRY is small. No, you should try to memorize those 4 numbers. And then you should know, okay, that's on the smaller side, so that means it's going to be mutated at a slimmer chance than the dystrophin gene. Okay, so yes, the globin gene would be an example of this, and they're homologous throughout their entire length of their sequence, like 80% similar. They only have slight variations. So here we have some of the globin genes. They're clustered together, so we said that they can be, or they can be dispersed on different chromosomes. So the fetal globin genes has a higher affinity for O2 than the adult globin genes. We want to be able to release that O2 into our tissues, so they're a little bit different, but they're still part of a gene family. So again, gene families arising due to gene duplication. So start with a single globin gene. Now we have it duplicated to alpha and beta. Then we have fetal and adult. So those are near identical and those fetal are near identical, and they're different than the adult, but they're still homologs. They're still doing the same function, but just a little differently. Again, same thing. Ancestral globin. Then we get down to all of these globin genes. We see, okay, 99.3%, 93.2%, super similar. Next. This is just that zoomed in. Next. Okay. So then he's saying, so nucleotides, we only have 4 choices, right? A, T, G, C. Well, proteins, we have 20 choices. So a lower percent similarity is more dramatic for amino acids because it's harder to get that similarity. We don't have to know the percentages, right? Like these percentages here, no, you don't need to memorize that. But I would know that they're like 80% similar for gene families. Just keep that like in your mind, they're like 80% similar. And then you don't need to memorize like any of these percentages. It's not going to test you on that. Okay. So we know that it's more dramatic for amino acids to be similar because it's harder for them to be similar. So even a smaller percentage of similarity is kind of good. So if 50% of amino acids, that's really good because more amino acids can take that place. So if we only have 4 options for nucleotides and we say that the nucleotide sequence is 30% homologous, we're like, okay, that's not that big of a deal. You only have 4 choices anyway. But if we say that the amino acid sequence is 30% homologous, then we're like, oh, wow, you can have 20 different choices there. So that's a higher percentage for us. That's all he wants you to know on this slide. Okay. Flat, basic linear alignment tool. It's longer with nucleotides and amino acids. BLAST is shorter and it's only nucleotides. We'll get into this later, kind of just introducing it. Okay, so gene duplication. We can relieve the mutational constraint by duplication. So if you only have 1 copy of the gene and it gets mutated, you're effed. But if you duplicate that gene and 1 gets mutated, then it's like, okay, we can start using our other copy because this one's like messed up. It's mutated, it's wrong. We can use our other copy. So gene duplication of important genes helps us relieve that mutational constraint. Okay, so here's a self-study slide, blah, 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 whatever. He says that antibody genes represent gene families with a slight twist because they need to be assembled. So if we go, okay, here's our variable regions. That's where the antigen binding occurs. I'm sure you guys know this because you're taking immunology. Okay, variable region in the light chain and the heavy chain. Okay, so this is the slide that I want to talk about. So we have 7 immune receptor genes that recombine during our life. Homology throughout and similar biochemical functions make the segments of which are these variable regions, part of a gene family. So these variable regions are all basically doing the same thing, just like a little bit differently. So they're all part of a gene family. The variable region is where antigen binding occurs. So it might be different antigens that are binding, but still antigen binding. So they're part of a gene family. Okay, this is kind of small, but I included the exam reading figure text legend. So we can go over this a little bit. So the cluster of the 5J gene segments is separated. This is kind of small. Let me try to zoom in. Okay, there. It's separated from the C region coding sequence by a short intron. Short intron right here, if you can follow my mouse. Okay, and from the 35 or so functional V gene segments by thousands of nucleotide pairs, which is why we have that little squiggle right there. So during the development of a B cell, a randomly chosen V gene segment, V3 in this case, so we're choosing this one, is moved to lie precisely next to one of the J gene segments, J3 in this case. So we're going to move this V3 to be next to this J3. The extra J gene segments, J4 and J5, and the intron sequence are transcribed along with the joined V3 and J3 gene segments and the C region coding sequence, and then removed by RNA splicing to generate our mature RNA molecule with our contiguous V3 and J3 and our C coding sequence. So then these mRNAs are translated into the light chains. Okay. Heavy chains have VDJ, light chains only have VJ. Introns between J and C regions are both in heavy chains and light chains. So basically, the point we're trying to make here is that if we go back to oh, come on. If we go back to where he said there's a slight twist because they need to be assembled. So we're going through this like splicing to move this B3 next to this J3. Then we're going through our transcription, or this is being moved next to this J3 transcription and then splicing to get rid of all those unwanted pieces. And then that's how we get our mature RNA. Okay. So gene families, functional definition. All proteins representing a gene family have a closely related biochemical function. So know these examples, that globin proteins are gene family, are an example of gene families, and they carry oxygen. And then all immunoglobulin proteins bind foreign pathogens, and they're an example of gene families. Know those two examples. Okay, supergene families. So they're homologous only through a section. So we said if we go back, oh, come on. So if we go back to this slide, we said gene families are homologous through their entire sequence, like 80% similar or more through their entire sequence. So then now we're talking about supergene families. They're only homologous in a section, like half. So we can see here that gene 1 and G2 are homologous throughout like half. Then they're completely unrelated or divergent region in the other half. Can you explain the main takeaways from the figure text? I'm not really sure what, yeah, so he just wants you to know that these are being put together. So these V segments are part of a gene family. They're being put together to make this immunoglobulin. So they're a little, they're like gene families with a twist because they're being assembled. So we're choosing which ones to pick when we're making our antibodies for certain pathogens. And that's all. That's why this is like the only highlighted thing. So these V gene segments are part of the gene family, and they can be kind of separated. So we know that gene families don't have to be like all clustered together, and they're being put together. Okay. So again, Supergene families only homologous through section, gene families homologous throughout. So then the gene family of immunoglobulins is a member of a supergene family that includes other gene families that encode for surface molecules involved in immune cell adhesion and recognition. So supergene families can have a physiological relationship without having the same biochemical functions. We can see that better on this slide. So immunoglobulins are a member of a supergene family that include surface molecules involved in immune cell adhesion, right? So our immunoglobulins are part of a super G family with T cell receptors, B cell receptors, class 2 MHC, class 1 MHC, and FC receptors. So all of these things have a similar physiological relationship, immune system, immune function, but they're not all having the same biochemical functions. So they have different functions, but they're still part of the immune system. All of the immunoglobulins are binding our antigen, and then we have all of these different receptors and proteins and stuff that are still functioning in our immune system, as you know from taking immunology, but slight difference in the biochemical function. So all of these are considered supergene family with immunoglobulins. So to compare and contrast them, gene family, all proteins in a gene family have closely related biochemical function. Supergene family, most proteins in a supergene family are part of a related physiological system, for example, immune system or neuronal tissue. Are there any other supergene families that we need to know? I would just know like the ones he goes through here. So all of these immune system related proteins and receptors, and then he gives you another example of neuronal tissue. He's not going to test you on something that isn't in his lectures. So are immunoglobulins both part of gene families and supergene families? Yes. So genes can be a part of both. They can also be a part of gene clusters. They're not exclusive to a gene family or a supergene family. So like we said, immunoglobulins are binding our antigen. So immunoglobulins together are part of a gene family. Immunoglobulins with T cell receptors, MHC class 1, MHC class 2, B cell receptor, etc., all of the other immune functioning proteins, that's supergene families. Then if we look back on this slide, we can see like, oh, okay, these V segments can be part of a gene cluster because they're all closely located on the same gene. So they can be all of those things, gene families, supergene families, and gene clusters. Just depends on what you're relating them to. Okay, so now we're talking about a gene cluster. So genes that are physically together doesn't necessarily mean they're automatically related. So gene clusters are composed of homologous genes encoding proteins with a closely related molecular function, or it can be, so they can be genes that have closely related biochemical functions like those V variable segments, or it can be another gene is part of a gene cluster, but they have a close related molecular function, not necessarily the exact same biochemical function, which is why we're saying they're not automatically related. So gene clusters range from extremes where duplication has generated two adjacent related genes to cases where hundreds of identical genes lie in a tandem array. So basically what we need to know here is that genes may have the same function, but the term gene cluster does not imply gene family. Gene cluster is more of like a physical term that shows genes are close together. So gene cluster means that they're close together. Gene family means that they have related biochemical function, and supergene family means they're part of a related physiological system. Okay. So here we can see our class 2 MHC genes are part of a gene family. So then we could say like these genes right here are part of a gene cluster. And then we can say this HLA complex is a supergene family. If that makes sense, that can show you like all the relationships. So there's other genes that are stuck in here that are not necessarily part of the gene family, but they can also be part of a gene cluster because they're right next to each other. Do I mind repeating that explanation? For which question or for which topic? This slide or the previous slide? This slide. Okay. So if we have our class 2 MHC genes, that's a gene family because they have a closely related biochemical function. So then we can say this segment right here or this segment right here with the gray spaces in between is a gene cluster. So this HLA, DR, beta, beta, alpha, and then the green spaces in between can be all part of a gene cluster. But these gray spaces in between are other genes that are just like stuck in there. They're not necessarily part of this class 2 MHC gene family, but they are part of the gene cluster because they're right next to these genes on the chromosome. And then this class 1 MHC gene and class 2 and class 1 MHC gene can be part of like a super gene family. Does that make sense? Okay.
Hi, Ivy.
Yes.
So the only part that I'm not understanding is which one is the super gene family? Like, is it the entire HLA complex or is it just each class?
So the entire complex can be part of a super gene family, and the class 2, class 1 individually can be part of a gene family. So it has to have similar biochemical function to be part of a gene family. So if I can give you another example, let's say we have multiple genes for our nervous system that encode for myelination complexes of our neurons. So those multiple genes that are all encoding for myelination will be part of a gene family. But those genes and other genes that encode for making the neurons themselves will be part of a super gene family because they're part of a related physiological system, which will be neuronal tissue. Does that make sense?
That made a lot more sense. Thank you so much.
You're welcome. Okay. Here is this slide again. We already went over that. So now we're going to get into this moderately repetitive DNA sequences. So you should know these characteristics. Like you should know it's roughly 100 to 1 million copies, and it's usually 200 to 15,000 base pairs in length, because these numbers are what's going to differentiate between the 3 different types of DNA sequences, the single copy, moderately repetitive, and highly repetitive sequences. So you should definitely know like these 4 things. So roughly 100 to 1 million copies, can't be moderately repetitive with only 2 copies. Usually 200 to 15,000 base pairs in length. That's a sharp upper limit for us. Then can be clustered or interspersed, like our RNA genes or our histone genes, and it may be coding or non-coding. So most of the non-coding is just junk, again, comes from viral genomes integrated into our genome, and there's no physiological role, but sometimes they can be important for structural purposes, like lining up our DNA during recombination. So here are examples of our moderately repetitive DNA elements. We have lines and signs. So lines stands for long interspersed nuclear element. And then signs stands for short interspersed nuclear elements. The difference between them is that lines are bigger than signs. These are kilobases. And since they're bigger, the size difference represents the difference for lines to encode a protein. They are larger. He said for us not to worry about this open reading frame stuff. So the fraction of the genome for lines is 21%, which is pretty significant. And there's two major points that he wants us to know. So we have relatively large repeating DNA interspersed around the genome that does have coding regions. So these are lines. They do have coding regions. And the most important one is reverse transcriptase, which we know is left over from viruses incorporating their genome into our genome. And they're expressed extensively in gametogenesis and germ cells. Then the second point he wants us to know is that we also have these short interspersed repeats around the genome, and those are our signs. And what's separating them is that the long ones can have a coding region, most importantly, reverse transcriptase, and the short ones do not. So that's our takeaway from this slide. And those are examples of our moderately repetitive. So you should know those examples. It might be helpful to make a chart and say, okay, examples for moderately repetitive are lines and signs, and then compare and contrast them. And then you can say examples for the gene families are globin and immunoglobulin. Then you can say, I think that's all our examples as of right now. Yeah. Because these are all still globin. Yeah, globin. Yeah, so make charts like that. And then these, we don't really have that many examples because almost all of our genes are in that category. So I would make a chart and I would include these globin and immunoglobulin as examples that he used for gene families right here. And then just know the difference between a gene family and a supergene family and a gene cluster. And then I would also write down lines and signs as an example of our moderately repetitive DNA elements. Compare and contrast them. Lines are bigger. They, yes?
So would gene families and gene clusters and supergene families be examples of the single copy DNA? Or
not necessarily.
Not necessarily. I was just wondering because he had it after
Yes.
that, so I wasn't sure if he was like, clumping that all together.
Not necessarily, because if we go to this slide now where we have multiple tRNA and rRNA genes, if those are right next to each other, then they're gene clusters. They're a part of a gene family because it's literally that same gene. So they're doing the same biochemical function. So I wouldn't say that they're only for that single copy.
Okay, thank you.
So how would you be able to tell the difference between a single copy DNA sequence and a moderately repetitive DNA sequence? You can tell the difference because moderately repetitive DNA sequences have roughly 100 to 100,000 or a million copies. And our Single copy is only one copy or a few copies. There's no upper or lower size limit, whereas we had an upper and lower size limit with our moderately repetitive. And he gives you the examples he wants you to know for your moderately repetitive, these lines and signs. And then for the clustered, he says the rRNA genes and the histone genes. So that's another example to add to these moderately repetitive. He's not gonna like give you a gene and say which one of these, like he's not gonna give you like a RB gene is which. If he's gonna test you on these examples, it's gonna be lines and signs are an example of blank. And then it's gonna be those choices, and then you pick moderately repetitive. Okay. So then now that we talked about our lines and we talked about them encoding for reverse transcriptase. Yeah, his slides are a little confusing. It's okay though. We'll get through it. His tests, I promise you, like he teaches you at a level where you're like, wow, he wants us to know all of this stuff. But then he tests you like super fairly. And I know like a lot of my peers last year said that they like overstudied for genetics, and when they got a question that they thought was too easy, they like overthought it and picked the wrong answer by overthinking instead of just being like, yeah, that question is just that easy. Like I studied hard enough that it's easy for me. And like my TA last year told us, if a question seems easy, that's because it is. Think about it like the person that sits in the back and only comes to lecture like once a week or something. Because he's not trying to necessarily trick you. He just like, if you studied the material, then you're good. Okay. So now that we talked about those lines and how they encode for reverse transcriptase, he's going to show us what that means for us. So here, this L1 is our line 1, and we're starting transcription right here. So since we start transcription right here by this L1, and we're transcribing this E3, which is an exon 3, it's going to be included in our RNA. And then we know that our lines can do reverse transcriptase. They encode for reverse transcriptase. So the primer is left out of this drawing. He's very adamant about making sure we know that the primer is left out of this drawing. So just remember that it's there. But we go now from this hybrid RNA using reverse transcription to this copy DNA. So now that this copy DNA exists, once it's double-stranded, it can insert itself back into the genome. And it doesn't have to be back at that same gene. So we started off at gene, oh gosh, what's happening? We started off at gene A, and now we're ending up in gene B. So it got transcribed into an RNA, went through reverse transcription, became this copy DNA. Once it was double-stranded, it inserted itself back into our genome, not necessarily at the same spot. So now we have this hybrid gene. And this exon 3 was just along for the ride. So now it's in the middle of a new gene, and this is how we get supergene families. So if this gene B was, let me try to give you an example. I'll stick with the neuronal stuff. So if this gene B was for all your neurons, and then we have this exon 3, which is for myelination. Now it's perfect that we have it on our new gene, which is for all of our neurons being formed, because when we transcribe this, now we can transcribe the gene that makes the myelination to go on those neurons. But they're not the same biochemical function, but they're the same physiological function. So it's beneficial for us to have it on that gene now. So we call this exon shuffling. So we shuffled this exon from gene A now to gene B. And we can move around the genome thanks to this reverse transcriptase over millions of years. So it was probably evolutionary favorable that once we make a new neuron, we got to myelinate that. So if we're transcribing all of our genes to make neurons, we're going to also transcribe this gene to make the myelin.
Ivy, I have a quick question. So I'm just kind of getting confused. Like, what really is the difference between lines and signs and exons and introns, respectively? Are they just specific exons and introns?
So exons are going to be the protein coding regions.
Right.
So these moderately repetitive DNA sequences may be coding or non-coding. So it's just a sequence that's repeating that can encode for this reverse transcriptase, but it's not like encoding for a protein that I need. Like if I need a transmembrane protein for my immune system, like my T cell receptor, it's not encoding for that. So it's not necessarily an exon encoding for proteins that we need, but it's not an intron that just gets cut out and not used. It's its own category.
So it's not even like a line wouldn't even be within an exon. It's its own place on the gene.
Mm-hmm.
Okay.
So like, for example, in this image, this gray stuff is our introns.
Mm-hmm.
Gonna get cleaved out. We don't need it. The E1 is an exon that encodes for like some protein. So if this E1, E2, and E3 were part of a gene family, sticking with my original example, they're all doing myelination of neurons. They might be doing them a little bit differently, but they're all doing myelination of neurons. So it's encoding for our proteins that are myelinating our neurons. Mm-hmm. And then here we're separated by more introns that are going to get cleaved out. Here is our line 1, so it's encoding for reverse transcriptase, and that's allowing us to pick up this exon 3 and move it to a new gene. So it's like its own thing.
Okay. All right. That makes more sense. Thank you so much.
You're welcome. Okay, what exactly are lines? I know they're long interspersed nuclear elements, but what do they actually do? They encode for reverse transcriptase and allow us to complete this exon shuffling. That's what he wants you to know about these lines.
Hey, Ivy.
Yes.
Just going off what Lance was asking about, because I'm not sure I understood. Are lines and signs determined only by exons?
No. Lines and signs are their own thing. It's whatever sequence, and they're just repeating at this rate, usually 100 to 1,000,000 copies. They're its own repeating sequence.
And that could include introns.
And that what?
That could include introns.
It's like, okay,
is it like a stretch of DNA that has, some functions due to its exons, but that stretch of DNA, the line or the sign, includes introns?
So if we look at this image, this can help me explain it better for you. So these are exons that encode for proteins that we need, right? So this stretch of DNA in here is not encoding for proteins. That's where we can see these repeats in this stretch of DNA. But you can see that we still have other stretches here. So those are the introns in between these signs. And you can see that this stretch of DNA in between here can also be kind of considered an intron in between our exons. But we won't necessarily get these like cleaved out or spliced out. But you're not going to see these repeats within an exon. They have a different function. So the line function is going to be for reverse transcriptase. The sign function, we don't really get into. I don't even know what signs are for. They're just repeats. And it's not necessarily the exons determining their function, because the lines function and the signs function in their own way. So lines have reverse transcriptase. Lines can take an exon. So like if if we evolve, if it's evolutionary favorable for this exon 2 to be on a different gene, if there's, here we see a line right here, that line, well, there has to be a line before it. So if there's a line before it, we can go through transcription, and then we can do reverse transcriptase and insert that exon 2 into another gene. Does that answer your guys' question?
Yeah, I'm gonna have to sit on it for a little bit though. Thank you.
Okay. Okay, hold on. The chat. Line 1 is transcribed normally 5' to 3', and then once it's considered hybrid RNA, it uses reverse transcriptase to make the hybrid cDNA. Yes, that is correct, Adam. Exons code only for proteins, while lines encode for other things such as reverse transcriptase. Yes, that is correct. Is it exon, mRNA, reverse transcriptase, new copy exon? So you guys have to remember that we're just working with a DNA sequence. All of this is just a sequence of DNA. So our lines and signs are repeating. For example, they could be repeating ATC, ATC, ATC over and over again for 100 to 1,000,000 copies. So we can have 100 copies of ATC, 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 and then we can consider that a sign. If we have like 1,000,000 copies, then we can consider that a line. And since it's bigger, that can encode for reverse transcriptase. And whatever exon is next to that line in the DNA, it's like along for the ride. So this whole thing right here is just a stretch of DNA. So this sequence could be like GCAT. That's our exon 1. That's going to encode for a protein. And that protein has its biological function. Sticking with my example, it's myelinating neurons. So then now we have some introns here. Okay, we don't need this sequence apparently. So then now we get to our exon 2. That's encoding another protein, which biological function is myelinating neurons. Then we have these introns over here. Okay, we got it. Now we get to this line 1. So if we want to transcribe this line 1, we would start with transcription here. And it just so happens that this exon 3, whose biological function can also be to myelinate a neuron, is along for the ride because it gets transcribed along with this line 1. So all of this is just one sequence of DNA. So then once we go through transcription, now we have our line 1 and our exon 3 that just got taken along for the ride. And then this is our RNA. So then now we're going to go through reverse transcription to make a copy DNA. It's going to insert itself back into a different gene. So these lines and signs are not taking place within coding regions for proteins. That would mess up the protein code. They can occur in these stretches that we have here of non-protein coding DNA. So if we look here, we see, okay, we have some signs in this stretch, okay, but we don't have any signs here because this is our protein coding region. We have lines here, not in the same part of signs because they're different, not in our protein coding region. So let me check the chat again. Do lines only encode reverse transcriptase, or do they encode other proteins and enzymes? He said that does have coding regions. The most important we're going to talk about is reverse transcriptase. So for our purposes, reverse transcriptase only. Lines and signs have their own place on the gene as well as their own function, just as a promoter gene would. I know blank doesn't like that. Yeah, they have their own function. They're their own thing. It's their own sequence. It's not necessarily due to an exon's function. The hybrid RNA in the picture is mRNA, correct? Or did I mix it up? That's going to be like our primary transcript. So it's not like finalized because it's going to go back through reverse transcription and become copy DNA. It's strange to me that a sequence can be expressed without being an exon. It's not making a protein. So I don't, it's getting transcribed. It's doing reverse transcription. So yeah, it's making reverse transcriptase, but it's not like encoding for like a protein to make like a transmembrane protein for a T cell receptor or something. I know this concept seems like a little difficult. It was difficult for me when I went through, but let's move on. And if you have any more questions, I'll try to answer them at the end. Again, I don't have a PhD in this stuff. I'm just telling you guys like what's important to know for your exam. So the RB gene is a huge gene, and it leaves room for lots of repeats. Okay, so here's the room for our repeats. And like I said, these repeats are not happening within coding regions. So here we have a lot of coding regions, and we see like we don't have any repeats going on. And he said previously that the coding region is denoted with these boxes of exons. So he gave you an exam question for this slide, and it said, which of the following strands is the template strand for the primary transcript of the RB gene? It's going to be the bottom. Because if these are what we want, we have to do the other strand for our template strand, because we're going to use the template strand and synthesize the complement to the template strand, which is going to be these genes up here. Okay. So all genomes contain multiple tRNA and rRNA genes. And that's the case because these genes need to have many copies since they only get used one time to make one ribosome. It's not like we can have that mRNA and make multiple copies. We're making one ribosome. So for example, the globin gene only has 3 or 4 copies, and for this rRNA gene, we have 280. So the take-home message is because our RNA can only be used one time to make one ribosome, we need to have many copies to keep up with our demand. So this is our gene. The black is our exons. So we're looking at this exon. We're going to make our 18S, our 28S ribosomal subunits. So those are our protein coding regions, our exons, and then we're going to have these like little sequences in the middle here. So when our 18S and 28S are cleaved out of the primary transcript, once they're cleaved out, we're left with RNA that we don't need. So we call these IVS. These IVS are intervening sequences, and they're distinct from an intron because we're not splicing together the exon of 18S and 28S because they're being made into separate pieces, separate subunits for that ribosome. They're separate pieces. They're not going to be spliced back together. They're going to be processed separately. So if they're processed separately and they're not spliced back together, those that RNA in between them that we don't need is not technically an intron. It's an intervening sequence. And I remember that this was on the exam. So you have to know that intervening sequences are not introns. And it's when we have this 18S and 28S cleaved out that they're not being spliced back together. And that's how we're creating these intervening sequences 1 and 2. Does that make sense? If you have a question, stop me because that's important to know.
Do we need to memorize the 18S and 28S?
Yeah.
Or there's the ribosomes, I think.
Mm-hmm. Yeah. So you
memorize
shouldn't,
those,
yeah.
Okay.
Hey, Ivy, can you go over this slide again? Like, just really briefly sum it all up.
Okay. So these are different subunits for our ribosome. You know that a ribosome has two subunits. So it's our 18S and our 28S. So when we want to make our 18S and our 28S for our ribosomes, we're going to go through transcription, translation, whatever. But when we go through transcription, we transcribe all of this. It's our RNA. Okay, we're not going to splice together the 18 and 28S. So you know how we cut out our introns and then splice together all those exons? We're not doing that in this case because they're being processed as separate pieces. So if they're not getting spliced together, then the introns are not called introns. They're called intervening sequence. So you can think of this like kind of similar to an intron. It's what we don't need when we have our exons, but they're not getting spliced back together, so they're not technically an intron. They're termed intervening sequence.
Well said. Thank you.
Mm-hmm. Okay. Are 18S and 28S still considered exons? Yes. To my knowledge, yes. Did someone else have a question?
Hi. This might seem like a silly question, but in 2 slides before, when we had the RB1, those different at the top, the 1, 2, 3, 4, 5, 6, all that. In a normal situation, because the one that you just had said that it had IVS, the intervening sequences, will these be all put together? Will they all be put into one protein at the end? The 1, 2, 3, 4, 5, 6 at the top here, the RB.
I'm not sure. Depends on, hold on, let me see. RB1 gene. Yeah, they could be. Because if we're looking at, for example, oh, come on. If we're looking at this, for example, so this 1 exon, 2 exon, this is all one gene. So it can have many exons getting spliced together to make one functional protein.
Okay, and if he were to ask us any question about, like, if you were to give us a picture or anything like that, you'd have to specify the intervening sequences then to make sure, like, you know, one whole protein.
So he wouldn't ask it to you like that. He would just more so ask you, like, he would more so ask you, IVS1 and IVS2 are considered introns. Yes, like true or false. And then you would say, no, they're intervening sequences. Or you would say, like, it would be something like 18S and 28S create introns. And you would say, no, because they're intervening sequences. He wouldn't necessarily, like, show you a picture of this and ask you to say, like, oh, what's that gray? Is it an IVS or is it an intron? He's not going to ask it to you like that.
Okay. Right. Thank you so much.
Uh-huh. So IVS1 and 2 are the black parts? No. The black parts are the 18S, 28S. See, like, these black parts here are our exons.
Wait, so the gray parts are the IVS then?
Yeah, but he's not going to show you a picture of this and ask you to, like, designate which one is an IVS. But, like,
yeah, I just wanted to visualize it for myself. So the 2 black pieces in the middle, those are the 18S and 28S. Or I meant the IVS and the IVS1 and IVS2. And the gray parts are the subunits.
The gray parts are the IVS1 and the IVS2.
Oh, okay.
Mm-hmm. What is 5.8S? It's another ribosomal subunit that wasn't included in this chart, and he said it was a shame. It's like not that important for us to know at this time. It's mostly going to be knowing that this 18S and 28S are not getting spliced together. They're getting processed as separate pieces. Okay. I don't know what the ETS is. I'm sorry. Okay. So we talk about the demand for histones. Whenever we replicate our DNA, if we create a new daughter cell, we need a lot of histones. Again, we only have 150 base pairs of DNA wrapped around a histone. If we have like billions of base pairs of DNA, that's a lot. So we need a lot of histones, meaning we need a lot of histone genes. So histone amplification is having many repeats. So we have all those genes. So we can generate a lot of mRNA, and then each mRNA can be used many times. So not only are we generating many copies of our mRNA, we're going to also use that mRNA a lot, which makes sense because we need a lot of histones. That's the takeaway from this slide. Okay. VNTRs. I kind of like VNTRs. They're cool to me. You don't need to memorize any of these sequences. Don't worry about that. Don't waste your time with that. So VNTRs are variable number tandem repeats. They're relatively small numbers of nucleotides, and they get repeated a differing number of times. And they're the basis for like DNA fingerprinting. So they're highly unstable compared to the stable lines and signs, which is why they can be used to identify individuals. So lines and signs are like fairly similar for every person, but these VNTRs are different, which is how you can use them to identify like a DNA sample. So this is what they use at crime scenes. If somebody left DNA at a crime scene, we can use their VNTRs to match it to that individual. So we went over PCR multiple times now. So now we can like apply it to something. So, and then also this STR is just a subclass of VNTRs, but on the exam, he will use VNTR if he's asking you about it. So don't really worry about this STR. So if we want to know the sequence, we can have these primers. So we look it up on the genome browser, whatever, and we find a primer for it. So we can use that primer. That primer pretty much works for like everyone. And then we see the sequence that gets made with those primers. We see like we're duplicating our sequence. We know how PCR works. Okay, now we can work with this DNA. So the sequences can be predicted for both sides. And then we'll see like how many repeats they have. So like I might have it repeating 10 times and another person might have it repeating 20 times. So, all right, we know how PCR works, right? We have our DNA that we're trying to amplify. We put our primer, we have our DNA that we're trying to amplify. We can use helicase to separate them. We put our primer down. Like you said, we can use Genome Browser to find the sequence that we need for the primer. We use DNA polymerase, synthesize those nucleotides. We get a double-stranded DNA molecule. Again, then we separate and do it all over again until we have many copies of the DNA that we want to synthesize. Okay, this again, just isolate and amplify. I want to talk about this first for a second. So here we can see that the individual has this repeat for their STR1, STR2, STR3. So you can see how that's different from individual B. Their first one is kind of flipped. Their second one is smaller, and then their third one is longer than individual B, right? So everybody's going to have different numbers of repeats for these VNTRs. So if we count like these little nucleotides, like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, for example. So they have 10 repeating on this chromosome versus that's like a lot. I'm not counting all of this. So we can use that to build a DNA profile. So like we take one pair of, he hates that, but one pair of homologous chromosomes, and then the next, and then the next. So we can build like a DNA profile. So the odds of one person having the same number of VNTRs for all of their chromosomes compared to another person is like super low, and that's the basis of DNA fingerprinting. So then you can see like the forensic sample matches B and it doesn't match C or A. Okay, so now to get to this gel electrophoresis part. So what they do for this forensic sample is take the sample and amplify it using PCR. So then now let's talk about gel electrophoresis. So it's a semi-solid matrix right here, which impedes the travel of molecules through the matrix. So the big ones go slowly and then the small ones go faster. The anode is at the bottom, the cathode is at the top. So DNA is going to want to travel to the bottom because DNA is negatively charged. So it's going to want to go to its opposite charge, which is the positive on the anode. So that's what they did here. The larger ones are at the top, the smaller ones are at the bottom. So number of repeats, 30 versus 5. So these VNTRs are just the variable repeat. Does anyone have a question on VNTRs?
Hi, Ivy.
Mm-hmm.
You said the VNTRs are unstable. Can you clarify what you mean by that?
Yeah, so everybody is going to have a different number of repeats. So if the sequence is this CATA, for example, I might have 4 repeats of it and you might have 20 repeats of it. So that's what I mean by unstable versus like these lines and signs. If I have 20, then you're probably going to also have 20 because they're stable for the human population.
So just like inconsistent?
Yeah, which is why we're able to use them for DNA fingerprinting because it's unique to each individual.
Okay, thank you.
So the probability of somebody having the same VNTRs throughout their entire genome for each chromosome is very, very, very low to somebody else having the exact same number of VNTR repeats throughout their entire genome.
Hey, Ivy.
Yeah?
Because it's from a maternal and paternal chromosome, would siblings have more likely to have similar VNTRs?
Yeah, exactly. So like you get some from your mom and some from your dad and stuff. And that's why if you are familiar with like DNA testing for like crime scenes and stuff, I know there was an example of like one woman submitted her DNA for like ancestry.com or something. And it was a match, partial match for something in CODIS. And it was like her uncle had committed a crime and they were able to figure out it was her uncle because they had a portion of DNA that was like similar. So these VNTRs can be similar for family members, but the probability of having it exactly the same throughout all of their chromosomes and their entire genome is not likely. So although they might have similar numbers of VNTRs, maybe for a few chromosomes, it's not going to be the exact same throughout their entire genome.
Thank you.
Uh-huh. Are VNTRs the same in identical twins? I'm not sure, actually. I'm not sure. That might be a good question to ask him, though. He might like that question. Okay. So those were our examples of our moderately repetitive DNA sequences. So now we're going to talk about highly repetitive DNA sequences. So this is at least 1 million repeats or more. So when we talked about our moderately repetitive, we went from 100 to 1 million. Now we're like at 1 million and up. And then it's 6 to 100 base pairs in length, and they're clustered. They're all non-coding sequences, and the examples that he gives us for these are telomeres and centromeres. They don't code for proteins or structural RNAs. So we know what the telomeres do. They add on junk DNA to our template strand to provide RNA primers to be laid down, and then DNA polymerase to finish synthesizing the lagging strand. And then we know centromeres are where the two sister chromatids get pulled apart. And our telomeres are preventing from chromosome degradation and fusion. And our centromeres where our kinetochore forms. And that's where we're going to stop today. So if you guys have any more questions, you guys want me to go over something else, like go over something again, let me know.
Hey, Ivy.
Yeah.
Hey, I have a question. It's not regarding like the material that we went over today, but it's about those articles that we have to read.
Mm-hmm.
That are mandatory. What would like your tips be for interpreting the articles or the takeaways from every article?
So you can use my notes as like an example of what you should be doing for the articles. I'll be taking notes on this second article as well and uploading them, but you should be reading those highlights. And I know sometimes it's like one word, so you have to read maybe the sentence before or the sentence after to understand the context of it. So I would just read through those highlights and then try to see if you can relate it together. So let me pull up the notes I took on the telomere syndrome article. So in the telomere syndrome article, the first thing we talk about is the dysfunction of telomeres in some degenerative disorders. And then it talks about typical manifestations. And then we know what telomeres are and what their function is. So then we know that dyskerrin is an essential component of telomerase enzyme making the telomeres. So in dyskeratosis congenita, we have a mutant telomere gene characterized by short telomere defect. It gives us those symptoms. So I would know like, okay, so we learned about telomeres. We learned about what their function is. This is what happens when telomeres are mutated and they're dysfunctional, and we get this disease, and it's caused by this protein being mutated in telomerase. These are symptoms like that. Does that make sense?
Yes, that makes perfect sense. So I was looking at those articles that you actually were talking about, not articles, those notes that you said that you uploaded. It's under modules, under genetics, right? Okay.
Mm-hmm. Yeah. Are the quizzes going to be reopened so we can retake them or review them for exams? So the quizzes should be, if you took those Friday quizzes during the allotted time that you were supposed to take them on Fridays, then the quiz is open for you. You can go back and look at it. So then after the review session, we go in and we show correct answers. So you should be able to see the correct answers. So it should be open for you.
Do you have any additional questions today or no?
I do. I was just going to wait and see if anyone wanted me to explain something else before I got to them. But if we're ready, we can get to them now. In reference to the practice exam that you will be posting, will the correct answers be available? Yes, I'm going to post the practice exam, and then I'm going to post the key. So you could take the practice exam, and then, you know, I probably will do it as like a PDF. I liked doing that because then I could write on the exam. So I'll post the exam and then I'll post another PDF with the key so that you don't see the correct answers when you're taking it. Okay, so if there's no other questions that you guys want me to go over right away, I can open up the practice questions for today, and then we could go back to the...
I have a question real quick.
Okay.
In regards to the Friday practice exams, are you guys going to reset those exams to where we can retake them, or no? Okay.
They're just going to be, that's how it was for us too. They're just open for you guys to like look at them. So what I would do is just like cover my answers. Or if you guys have iPads, if you open the quiz in the Canvas app on your iPad, you can just copy and paste it. So what I would do is copy and paste it, and then it doesn't say like which answers are correct. So what you can do is if you have Notability, you insert a text box, then you paste it there, and then if it says correct in the text, I would just delete it, and then I would have one as like my test copy, and then I would have one as a key. So I deleted all the correct, like sometimes when you copy and paste it, it'll say like correct next to the right answer. So I would delete that, and then on my key, I would just highlight them. Yeah, you can put them into a Word document too.
Okay, thank you.
All right. So then we'll do this. And if you still have more questions, you can just stick around after and I'll answer them for you. So as always, just put your answers in the chat. I'll give you a second to answer them. So what subunit of TF2D binds to the Tata box? Yeah, the correct answer is A, TBP, Tata binding protein. Okay. True or false, you are likely to get 2 mutations in one codon. Good, that's false. That's why if you have the first sequence that we talked about for valine, that was only different by 1 letter. That's why you're more likely to get a mutation from valine to glutamate than you are if you had 2 different letters, nucleotides. Members of a gene family are homologous throughout how much of their sequence? Good. They're homologous throughout their entire sequence. There's slight variations, so they're homologous by 80% or more. Okay. The globin genes are an example of what? So the globin genes are an example of a gene family. He gave that to us specifically. They all have the same biochemical function. So make sure you know that. True or false, genes in a gene cluster are related to each other. Oh, sorry. This true was actually accidentally highlighted. I will fix that, but the answer is false. They can, oh, whoa, what's going on here? Whoa, this got messed up. I'm gonna fix this before I post it for you guys. But the answer is false. Genes in a gene cluster are not necessarily related. They can be, or they don't have to be. They're just in close proximity on the chromosome. It's more of like a location identifier. So I'm sorry, I will fix that. Okay. Any questions on those before we move on to the next half of the PowerPoint?
Hi, Ivy. Can you go back to the second question?
The second question. This one?
Yes.
Mm-hmm.
All right, thank you.
Mm-hmm. All right. Let's go on to the next. So 100 to a million copies of a gene is a characteristic of what type of DNA sequence? Good, yeah. It's the moderately repetitive DNA. So a million and more is going to be highly repetitive, and single copy is going to be like one or a few copies. And you should know that for the exam. What is the most? Yes.
In the kilobase value.
What was that?
I thought we needed to know that in the kilobase value.
This?
No, yeah.
So
specific gene. No, that was the specific genes. I apologize.
Yeah, okay. Perfect. All right. So what is the most important moderately repetitive DNA sequence we talked about in lecture? So I see that some of you are getting this right. It's going to be the reverse transcriptase. So let me pull that up for you to show you what I was getting at with that question. So he said, on this slide, we have examples of our moderately repetitive DNA elements. We have lines that encode for our reverse transcriptase. So I could have put lines encoding for reverse transcriptase, yes. But reverse transcriptase in the lines is what gives us our ability to do exon shuffling, which is another big concept that we need to keep in mind. Okay. Of the two, which encodes for protein? Yeah, so that's going to be our lines encoding for our reverse transcriptase, which is what he said was the most important that he wants us to know. Is this the only way we have reverse transcriptase ability in the genome? That's a good question. I'm not entirely sure about that. I just know that that's one example. I don't know if that's the only example. That might be a good question to ask him. I'm not entirely sure. So true or false, IVS1 and IVS2 are not considered introns. You guys are saying true, and you are correct. They are not, because they don't. The exons don't get spliced together. They get processed separately because they're going to become separate subunits of our ribosome. Okay. Using a gel electrophoresis, which will be at the top, and what is the charge at the top? So the answer is going to be D. Our large DNA segments are going to be at the top because our matrix, let me pull it up for you guys. Our matrix is going to be semi-solid. So it makes it difficult for the molecules to travel through the matrix. So the heaviest and largest DNA will be at the top. And we can see that illustrated again here in this figure. So we have our number of repeats. So like this would be like 33 or 34 versus like 4 down here. And then we can see that our cathode is negative, which is at the top, and our anode is positive, which is at the bottom. And this is what allows our DNA to travel to the bottom, because we don't want to be next to that negative charge if we ourselves are negative charged. So we want to go to that positive charge. So a DNA will want to move down to that positive charge.
So when you say top, you're just referring to the wells, right?
Mm-hmm.
Okay.
So like at the top right here, we're going to have our negative charge, and we're going to have our large DNA segments.
Got it. Thank you.
Mm-hmm. So does anyone have any more questions for me? Want me to go over anything else?
Hey, Ivy.
Yeah?
Would small...
Hello? Can't hear you anymore.
Can you hear me now?
Yes, I can.
Okay. Would small DNA segments also be negative?
All DNA is negative because they have those phosphate groups. What I was referring to was the like the cathode versus the anode. So the cathode is a negative charge, which will be at the top, and the anode is a positive charge, which will be at the bottom. So that DNA will want to leave like from the top and go travel down to the bottom. Does that make sense?
Ivy, thanks for your time today, and I hope your kitty heals up well.
Oh, thank you so much. Yeah, this has been a really stressful week. Now that she's out of surgery, it's kind of just monitoring her all the time. So I'm kind of exhausted. So thank you for bearing with me today.
Of course.
If anyone has any more questions, I can stick around for a few more minutes. If not, that's all I have for you guys. You're welcome, guys. Are there any questions today from me? Like the practice questions that we just did? Quick question, what kind of questions does he ask about genome browser? So if I remember correctly, the pictures that we got on our exam are going to be like homologous chromosomes versus sister chromatids from a karyotype. He puts pictures of the genome browser. Sometimes he'll put pictures of like, maybe that's in like future exams. But if he's going to ask you about the genome browser, he'll put a picture and then he'll say like, he'll point to something and he'll say like, what is this? Or he'll ask the type of information that you can receive from each of the different types of browsers that you can use. Or he'll ask you to infer something based off of the image. Hopefully that answers your question.
Ivy, the only takeaway about signs that we should have is that they're too short to be coding, and they take care of they use the reverse transcriptase that's already there.
Yeah, they don't encode for reverse transcriptase. That's going to be the lines.
Right, and they just use the reverse transcriptase the lines. Okay, thank you.
Mm-hmm. Yeah, it does seem like a lot of random information. I thought that too, and I'm not gonna lie, I didn't pay attention to it as much as I should have during the first exam block. So I got a couple of those questions wrong. I mean, obviously, I still did well enough to become your guys' TA, but I just don't want you guys to like downplay it as much as I downplayed it, because I don't want you guys to miss questions that you don't need to miss. It wasn't like hard and that's why I missed it. It was that I thought I understood it, but it was more of like a passive learning thing than an active learning thing. So when we go over it, you guys like obviously like listen to me and listen to him and stuff if you want to rewatch it to understand what he wants you to know and how to use the genome browser. But then after that, I would definitely recommend going in there yourself and trying to like piece together all the information yourself or with your study group. I know it's kind of like weird because we're all online, but like maybe if you guys could go to the library or like meet up at Starbucks or something and go through the genome browser together and help each other through it, that would be very beneficial.
Hi, can you explain what you were saying about like nucleotides having like 4 choices and the proteins having 20?
Yeah, so obviously we know that nucleotides are only A, T, G, and C. But if we look here, we have all of these different options for proteins. So let me go back to the slide that I was talking about that on. Okay, here. So if we're going to say that a sequence, like a nucleotide sequence, is 20% homologous, that's not a lot because you only have 4 choices to choose from. So like at any given position, if I have an A, I only have 4 choices for that position on like another chromosome if we're comparing them. But if I say I have like 20 or 30% homology with like a protein sequence, that's a lot because I would have had 20 other choices to put a protein at that, an amino acid at that specific spot. Does that make sense?
Yes, thank you.
Okay, you're welcome.
Hi, Ivy. I know you probably already said something earlier in the session about like the genome browser. Is there like, I was just wondering like what kind of questions and like if I should play around with it a little bit today before you cover it in the next session.
Okay, yeah. So he already lectured genome browser yesterday. So if you felt like you understood it enough to play around with it before the TA session, that would be awesome because then it like you'll just it would just kind of be more of a review for you, like hearing it again and making sure you got everything. But yeah, there will be questions about Genome Browser. He'll pull images. He'll ask you about what information you can have from each different browser, like what information you can use each browser to obtain. He'll ask you to identify stuff on like a screenshot. So yeah, if you like felt like you understood it enough yesterday to kind of play around with it yourself like this weekend, then I would definitely do that. Or you can just like rewatch him and then come to my TA session and then try to play around with it after. But you definitely should feel comfortable enough to pick out stuff yourself, because if you can pick it out yourself and he puts a screenshot of it on the exam, you're like, oh, I already know what that is.
Okay, cool. Thanks.
You're welcome. How's my kitty? She's good. She had surgery on Tuesday. We picked her up yesterday. She had to stay overnight. She's doing better. She is on a lot of pain medication. She can't lick her incision, so she's not necessarily sleeping through the night. So I'm not sleeping through the night. And it's going to be like a little while before she can like kind of go back to normal. So we're all just doing the best we can in this house. But thank you for asking. All right, if there are no more questions, then that's all from me. And you can always feel free to email me. You can always feel free to come to my afternoon session as well. The regular TA session for next week is on Tuesday, as always. So yeah, but I'll see you guys tomorrow for Fridays. So yeah, I'll see you guys tomorrow. Bye, guys.